So if we are talking just about ribosomal profiling technique, it may help us to understand certain translational mechanisms, although it may not reveal all their aspects without involvement of additional techniques. Say ribosomal profiling is obviously very helpful in the identification of translation of upstream open reading frames. And because of that, if these upstream open reading frames under particular conditions, they change translation of downstream main open reading frame that codes for the protein, then we could do ribosomal profiling under these different conditions and look for the relationship between translation of upstream open reading frames and protein coding open reading frames. And it could help us to understand how this regulation occurs. In addition, with ribosomal profiling, because of its ability to detect a specific reading frame that is being translated, you could identify switches in reading frame that occur on mRNAs. And such switches could happen, for example, due to things like ribosomal frame shifting, say when ribosome changes its translating frames, but also you would discover them simply in case if two open reading frames are being translated, but these open reading frames translate different reading frames. And so then you will observe such a switch. And so ribosomal profiling would be able to tell you that such switch exists, but it may not necessarily be sufficient to tell you why exactly such switch exists. Another example of translational mechanisms is, say, stop codon read-through, in which case what you may look for is ribosomes downstream of the stop codons, and you may detect them. But again, it can give you an idea that you may observe stop codon read-through there, but you wouldn't be absolutely sure about it, because it is also possible that there is a minor RNA variant that overlaps with the region of this stop codon, and in this minor variant, you have a translation from a different start and may not have stop codon there or translated in a different frame. So very often ribosomal profiling could give you ideas, some information that can push you towards identification of particular mechanisms, but it may not be sufficient for deciphering the details of these mechanisms.